I'm going to go ahead and swear you in. If you please raise your right hand. Do you swear or affirm that the testimony you'll be giving in these proceedings will be the truth?
I do.
All right. And then for the record, can you please give us your first name and your last name and spell them both?
My name is Sabrina, Sabrina Seehafer, Seehafer.
Great. Thank you. Mr. Kodiaga.
Thank you. What do you do for a living?
I'm employed by the State of Iowa, Department of Public Safety, Division of Criminal Investigations, Criminalistics Laboratory, better known as the DCI Crime Lab.
So we've heard a lot from other DCI Crime Lab people you work with, like the lady who just left over here, you work with her, correct?
Yes, I do.
What do you do at the Crime Lab?
My primary assignment is to the DNA section of the laboratory.
Can you briefly tell the jury your qualifications in terms of education and professional experience to be in your position?
Yes, I have a bachelor's degree in biology from Westminster College in Fulton, Missouri, a master's and a PhD in biochemistry from University of Rochester in Rochester, New York. Once I completed my graduate work, I was employed by the Iowa DCI Crime Lab. I started my training there, and that meets all the guidelines as set forth by the FBI Quality Assurance Standards. Since I've been employed at the DCI, I've also had over 500 continuing education hours, specifically in forensic DNA analysis. In addition, I'm also certified by the American Board of Criminalistics in molecular biology.
We hired an expert on DNA yesterday, so I'm not going to ask you much more on what DNA is. I believe it's still consistent to that. So we'll move on. Did you do DNA analysis in this particular case?
Yes.
What items, can you describe to the jury the items you received and what you did with them?
Specifically, I was submitted various items of clothing and shoes and some pieces of carpet, also some swabs and a known reference sample. A subset of those samples I did not analyze. The other ones, I would have looked for the presence of bloods, specifically looking for any red-brown stains on items that had not previously been screened with Blue Star from the crime scene team. I would have then done my screening test, presumptive tests to indicate the presence of blood. If they screen positive, they would move on to DNA analysis. If they screen negative, I would not take those samples on further.
Okay. Let's go back and let's talk first on when you received these items. If you had, did you write a report in regards to this case?
Yes, I did.
Do you have a copy of your report?
Yes, I do.
Will it help you to go through these items line by line to be able to testify in a more clear way for the jury about what you did?
Yes, it would.
The court's permission will ask that she uses a report to refresh her memory.
Any issue with that, Mr. Hawbaker?
No objection to that,
All
Judge.
right.
Your Honor, may I open my binder?
You may go ahead and you may use your report.
So starting with the items that were delivered to you on or about May 7th of 2024, can you tell the jury what you had and what you looked at?
So the items received by the laboratory on May 7th were a pair of jeans and another item that contained 2 sweatshirts and a long sleeve t-shirts.
Did you know who these were, or were these items belonging to a person by the name of Huston Danker?
That's what was indicated on the receipt form when I received the items for the jeans. It did not in the description indicate who the 3 sweatshirts belonged to, or the 2 sweatshirts and 1 long sleeve t-shirt.
When you say you do DNA analysis, what type of screening did you perform on these items that you've just talked about?
On these specific items, I was screening for the presence of blood.
And how do you do that?
So when I'm screening for the presence of blood, I first clean my bench area, individually lay out each item of evidence by itself. I examine that item, so I'm looking for the presence of any red-brown stains on the piece of clothing. I would then circle those areas, and then I would collect a subsample of that by dampening 2 sterile swabs and swabbing just a little area of the stain, set those aside, and those swabs would be what I would perform my screening test on. We perform 2 color change screening tests. So I would put the both clear liquids on one swab, one on the other, and then put hydrogen peroxide that pushes the reaction forward. If my screen tests are positive for the presence of blood, one of the samples should turn blue, the other should turn pink. In this case, on all those items, there were 9 stains on the jeans that did not screen positive for the presence of blood, so no DNA testing was attempted. And then on the other shirts, there was actually no stains noted when I examined them, so no screening tests were performed or DNA analysis.
Okay. As you perform your analysis on these items, do you at that time have a known DNA sample of Huston Danker?
I did perform testing on the known buccal sample of Huston Danker. That was received later, submitted to laboratory, after I had already examined the items in my report.
Okay. So you at some point have these known DNA samples, but initially you're just detecting if you can find blood in these items that you've testified about, correct?
Yes, so his known was submitted at the end of May, where the other stuff was earlier in May.
Was there blue jeans that you tested that was, I believe, that's the item number 46?
Yes.
And you say blood screening was negative on that item?
Yes, in the 9 areas I tested.
Were there other items that you tested?
Specifically, not on item 46. Those were the only areas. Then I moved on to the next items, item 47.
Okay.
Item 47 contained 2 sweatshirts and a long sleeve t-shirt. I examined each of those items individually and detected no stains on any of those items. So that concluded my exam on item 47.
Okay. What other item did you test?
I tested item 52.
What was that?
That was a pair of black shoes.
Okay. Can you describe what those shoes were?
I would have to refer to my bench notes to describe them.
Okay. If I showed you what's been admitted as exhibit 30, exhibit 300. Sorry. Do you recognize these as those items?
Your Honor, may I get gloves out of my pocket so I can handle the items?
You may.
Yes, I do recognize these items.
How do you recognize what's been admitted as exhibit 300?
So specifically, these black pair of shoes, you can actually see the DCI case number and the item number and the date I examined these 2 shoes written in silver marker, Sharpie marker, on the outside, just above the sole, on each item of shoe, indicating that this is the actual pair of shoes I examined that is associated to item 52. In addition, I also collected swabs of the soles of these shoes, and you can see these swab boxes indicate the DCI case number, the item number, and a description of where I swabbed, and bearing my date and initials of examination.
Thank you. What were you looking for in what's been admitted as state 300?
I was looking for the presence of blood, so once again I was examining them to see if I could find any red-brown stains.
And what was the result of your examination?
Screening did not indicate the presence of blood on 4 areas of the right shoe, and screening did not indicate the presence of blood on 5 areas of the left shoe. Therefore, no further DNA testing was attempted. You will note on those shoes, you'll see circles with a negative/negative. Those are indicating the areas in which I did my presumptive screening tests on.
When you examine items at the lab, do you always find presence of DNA or presence of blood or presence of what you're looking for?
No, we do not.
Okay. Is it a misconception out there that you as scientists will always have to find that?
Yes, it is a misconception. We have as many cases that don't have a presence of a body fluid that do. We also have a good subset of cases where no DNA profiles are developed.
And what are some of the reasons why sometimes there's nothing that you find when you test these items?
There could be a lot of different reasons. So one, there could never have been any biological fluids or DNA left on the item. Obviously, wearing pieces of clothing, we'd expect some small amounts of DNA. It could have the biological fluid or DNA could be cleaned off or removed. DNA also cannot be seen by the naked eye, but it can be washed off. It can be degraded, such as leaving something in sunlight, different nature factors, such as the dirt, and different things you spray things with can degrade the DNA and make it in a form that it's such small, tiny pieces of DNA that we're not able to develop a DNA profile from it.
Okay. So in other words, things like blood can be cleaned by someone, correct?
Yes.
Passage of time in terms of if this shoe that you're collecting is 3 years down the road or 2 years down the road, that passage of time can degrade that sample of blood that would have been on that item, correct?
Yes. As a piece of shoe or an item of clothing ages out, if it's a small amount of DNA or it's cleaned up, it wouldn't be present there for us to detect with our testing.
Were there any other items that you tested?
Yes, item 53.
Can you tell us about that?
Item 53 was a white rug. Once again, I looked for the presence of any areas of possible blood. My screening test did not indicate the presence of blood on 13 different areas of that rug, so no DNA testing was attempted.
What about item 54?
Item 54 was a large piece of carpet. So specifically in item 54, I did not do any blood screening tests for this. It was indicated when it was submitted in my communication with the crime scene team that collected it, criminalist Jonna Berry, that they had already applied a blood screening reagent at the scene when they collected these items called Blue Star Forensic. Because Blue Star Forensic uses the same portion of the blood as my other screening test, it was determined that I would not do any screening tests and I would just attempt to sample the areas where she saw the Blue Star reaction in that segment of carpet. So she helped me identify those areas, and I took cuttings of 6 of those areas and attempted DNA on them.
And what were the results of your findings?
No DNA profile was developed from 5 of the areas on the carpet. One area did produce a few DNA factors, but those DNA factors were just too weak for any conclusive interpretation.
Okay. And from your understanding, this was they came from a house where a person by the name of Huston Danker was supposed to be living at, correct?
Yes.
Was it surprising? Is it surprising to you that even though this person lived here, you still were unable to find DNA factors or able to retrieve DNA factors from these tests?
No, it's not unexpected. Carpet is something that doesn't either holds a lot of DNA or very little, and typically people walk on it with shoes and not your bare feet, so the bottom soles of your shoes, unless you walked in a biological fluid, would not have very much DNA present, so it's not unexpected. In addition, all of our screening tests are only indications of blood, not human blood. So if those areas did screen positive with Jonna's for blood, it could also be blood of another animal, where my DNA testing is specific for human DNA only.
Can you tell us about the other items that you tested?
Yes. The next item I examined was item 56. This was swabs from marker E and from marker B. Once again, my communication and the receipt form indicated that these swabs were swabs of area that had blue star reaction positive. So therefore, I did no further blood screening, but I did attempt DNA on them, and no DNA profile was developed from those swabs.
And again, we're talking about these items that were retrieved by Jonna Berry, who just testified before you, correct?
Yes.
What other item did you test?
I also tested a swab indicated as swabs of marker C. Once again, no DNA profile was developed from that swab.
What item did you test next?
I tested item 58, which was a pair of tan moccasins. No stains were noted in my examination, but they're indicated that they possibly saw something on the right sole that I didn't see in my examination at the crime scene. So I went ahead and swabbed the right sole area of that right moccasin to attempt DNA testing on it in case the stained area just lightened in the transport. And once again, no DNA profile was developed.
What's the next item that you tested?
Item 59, which was a pair of brown boots.
What were the results of your test?
Once again, no stains were noted on the boots, but I did attempt to swab the soles of those boots and do DNA testing. And once again, no DNA profiles were developed from the swabs of the soles of those boots.
And again, these were boots that, as reported to you, belonged to either Huston Danker or found at his house, correct?
These boots were collected by the crime scene team. As for the ownership, I'm unaware.
Okay. What else did you test next?
That was the end of my examinations of items from the scene. I then examined the known buccal sample of Huston Danker. So because you previously heard that DNA, if we develop a profile, we can't tell whose DNA profile it is. So I developed that known DNA profile so that if I did have DNA profiles developed, I would have that for comparison's sake.
Okay. And so you developed a DNA profile of Huston Danker.
Yes.
But fair enough to say that the bulk of your work that you did was testing most of these items and receiving a negative answer in terms of a DNA profile in these items, correct?
Yes, I did not develop any interpretable DNA profiles from all the items I sampled for DNA.
Even though they seem to be items that humans interact with, someone could have interacted with in terms of wearing the shoes or using the carpet in that residence, correct?
Yes, but my sampling of the shoes were on the outside area, so typically you would leave your DNA on the inside or the tongue area, so all of my testing was not in those areas.
So you talked about testing the outside. If you tested the inside, would you expect to find the DNA of the wearer of the shoe?
Yes, I would expect to get some DNA factors. When you're testing for wearer DNA on the inside of the shoe, sometimes it's not a complete profile, but I would have expected to get at least more than I got on anything else I tested.
And did you get anything? Or?
Only one sample, and it only gave me 3 DNA peaks, so I would have expected to get at least a partial profile or mixture from the inside of wearer shoes.
Can you explain that more?
So we're going to go back to what was probably talked about yesterday, so bear in mind that some of this might be repeat, but so because we test 21 locations, because you give it half your DNA from mom and half your DNA from dad, you have no possibility of leaving 2 peaks at any one location. So the 1 2-week profile from the carpet stain, I saw 2 peaks over the 21 locations and 1 peak at a sexing locus, a location. So in a wearer DNA, as you continually wear something, you can leave your DNA behind, but because it's skin cell DNA, it can be degraded or slough off. So that's why I said that I would at least expect to get a partial DNA profile consistent with a lot more factors than what I got from the one item, one cutting that I got a few DNA factors from. Sometimes because people share different items of clothing, you might get more than 2 peaks at locations, which would indicate a mixture of DNA.
I have no further questions. Thank you.
Mr. Hawbaker, any questions?
None, Your Honor. Thank you.
All right, thank you, ma'am. You may step down.